my screen shows you a summary of the physical and chemical digestion in the human digestive system, in which the first part of the diagram, the yellow part with yellow arrows, is showing you physical digestion and chemical digestion.
Right.
So you can see that every time when food is being physically digested, its main purpose is to increase the surface area so that that food can be very successfully mixed with the digestive juices. Now they become smaller in size, so the enzymes can also easily act upon them. Since they are mixed in digestive juices, so it allows the various mineral ions and other chemicals to get mixed in them, right? For example, the first case is that of, I think it's a piece of butter or fat or cheese, this one. Let me use my highlighter. So this one. And it shows
Yeah.
that it has been broken down by teeth into small pieces. And then they are still very big. Bile salts will break them down. Bile salts are actually found in the bile. Always remember, Hamza, that bile is made in the liver and stored in the bladder. Okay, made
Okay.
in the liver
Yeah.
and stored in the bladder. And bile is a secretion which does not contain enzymes, but its biological significance is that it contains bile salts. Okay, bile salts, and it's the bile salts that bring about emulsification.
Right. Okay.
Right.
Miss, I had a question about this thing.
Mhm.
So I was reading about the stomach, right? And I came across the glandular, I believe it's called the glandular tissue,
Mhm.
and it said that it secreted chemicals which are like such as hormones and then enzymes and proteins, enzymes in this case, which are pepsin, right? So when these are ingested, they act on the proteins which are present in the stomach. Now, there was another part that's mentioned that hydrochloric acid is also profound in the stomach, right? Now,
Mhm.
where does that hydrochloric acid come from?
It comes from the cells of the stomach.
So it's just produced inside. So there's like I read about it. There were three layers of tissues, right? There was a muscular tissue and an epithelial tissue, and then there was the glandular tissue. And
Mhm.
all of these, the muscular tissue and the epithelial tissue obviously do not secrete them. So they obviously don't. And the glandular tissue secretes chemicals, and
Mhm.
no hydrochloric acid is secreted from that. So
Mhm.
which part of it does it come from? Like which type of cell?
I think it's the
Um,
epithelial cells, as far as I remember.
epithelial cell.
I think so.
Okay.
Yes. It's actually the sodium and the chloride, so not sodium and chloride, hydrogen and chloride ions. They are secreted. In fact,
Okay.
I think we can find out the answer to this, but not in so much detail, because when we were reading this, wait, when we were reading about stomach,
Mhm.
mm, yes, see, it says, like all parts of the alimentary canal, the stomach wall contains goblet cells which secrete mucus, right? Acha, goblet cells, wherever they are, they secrete mucus. We have goblet cells in the respiratory system also. Then it says it also contains some other cells which produce enzymes and others which make HCl. The enzymes produced in the stomach digest proteins, so they are proteases. So this means that there are various varieties of cells inside the stomach. Some of them produce enzymes and some of them make hydrochloric acid. So must be the epithelial cells,
Okay.
right?
Right.
And one more thing. I still remember we had read something else as well. Wait a minute. Okay, fine. Yes, this is what we read. And God, I found it. It says, I'm reading from here.
Yeah.
Pepsin is an enzyme secreted from the stomach walls in a liquid called the gastric juice. And the gastric juice also contains HCl to kill harmful microorganisms. So this means that if we use the word gastric juice, it means it's a juice which is a combination of the enzyme and HCl. So both these are actually produced by the cells of the stomach, and both these, this one and this one together, make up what is known as the gastric juice. So this concept should also be clear.
Right.
And whenever you use the word, now see, proteases is a general word for enzymes that can break down proteins. But there are different kinds of proteases inside your digestive system. In the stomach, the name of the protease is pepsin. And in the duodenum, the name of the protease is trypsin. Okay. So you should remember that although both of them, they have the same job, but pepsin is found in the stomach and trypsin is found in the duodenum.
Okay.
Pepsin has an optimum pH of 2. Trypsin has an optimum pH of above 7. So this means the
So
optimum
trypsin.
pH of pepsin is acidic, whereas the optimum pH of trypsin is alkaline. So this is also a point to be remembered.
Okay. Okay.
Yes, I was talking about the bile salts, repeating once again that bile is a juice made by the liver, stored in the gallbladder, devoid of any enzymes, but rich in bile salts. The very reason that bile salts are there is that they will break down large drops of fats into smaller ones. And now it becomes easier for the enzymes to act on these small fat droplets. Look here. These were large droplets emulsified by bile salts into smaller ones. These are small fat molecules now. So that it becomes easy for the lipase to break them down into glycerol and fatty acid, right? This concept map is really good. And then you have, let's say, fish. Fish is rich in proteins. Water in the digestive juices dissolves some food. And then small pieces of food and some food in solution. So we have these polypeptide chains. And now the proteases, two proteases, pepsin in the stomach and trypsin in the duodenum, will break down these long polypeptide molecules into amino acids. Always remember, Hamza, that the protein digestion begins in the stomach. It is incompleted in the stomach and it is completed in the duodenum. When I say incomplete, I mean that the proteins will be digested to polypeptides in the stomach, whereas the polypeptides will be digested to amino acids in the duodenum. So also remember this.
And in the ileum, what do you call absorption takes place, correct?
Where?
In the ileum. Yes.
In the ileum. Digestion can also take place and absorption, both.
Okay, so digestion and absorption. So in the ileum, right?
Yeah.
Absorption obviously takes place through the network of capillaries. And Mr. was this word mentioned, which is I forgot. It was related to the lymphatic system. It was
Lacteals?
something like that. What was
Lacteals.
it? Sorry?
Lacteals.
Yeah, I believe it was that.
Lacteals. This is the
Actually.
spelling, lacteals. We'll do that. We'll do the structure of the villus because that's a part of the syllabus. So here they're focusing on the digestion of fat, here on the digestion of protein, and over here the digestion of carbs. So you can see that we have this complex starch molecule, which needs to be first broken down to maltose, and we need to further break down maltose to glucose. So basically, you know what this is? This is actually, this is a polysaccharide. So you can see how in different stages these molecules are broken first and from poly into disaccharide, and then from disaccharide to monosaccharide. Right? So starch to maltose, maltose to glucose. So we might say that if I ask you, is the digestion of starch completed in the mouth?
No, it's not.
Achha, in the mouth, starch is broken down by which amylase into what compound?
Miss, you mentioned the enzyme. It's amylase.
Into
Maltose.
maltose. And then maltose is broken down by what enzyme?
Maltose is broken down what enzyme? It's broken down by maltase.
Into
Glucose, miss.
okay. Can the conversion of maltose to glucose take place in the stomach?
No, it cannot.
So where does it take place then?
In the duodenum.
In the duodenum. Okay.
And Miss, there was another question I had.
Yes.
So it was that in the ileum, it said something like the ileum would secrete some type of what do you call it, maltose, right?
Mm-hmm.
So the walls of the small intestine would secrete maltose.
The
Sorry,
epithelial cells.
maltase, the enzyme.
Yes, the epithelial cells. In fact, we can
In
go.
the,
Huh. Yes, in
and
the
then and the what do you call starch is like converted into what do you call maltose in two places. So one of them is the mouth, and the other one is the duodenum, right? However, maltose is converted into glucose in only the ileum.
yes. You can see the names of the enzymes over here. See, you have amylase. What are you doing, Hamza? There's so much tuck 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 awas.
I'm typing this.
Acha,
Yes.
typing. Amylase.
Is it loud?
It's quite loud.
I tried lowering the volume. I don't know why it's not working.
Oh.
Because I have this section of notes that I type during class, and then I rewrite them with my pen like after the the class,
Okay, okay.
so I
So
don't miss anything out.
it's okay, it's okay. Now see, you can see amylase over here. It's made by two places. It's made by the salivary glands in the mouth and by the pancreas in the duodenum. Then protease
Yeah.
you have in the stomach also. Now this protease is the pepsin and this protease is the trypsin. Okay, this one is the trypsin. And then again, lipase is also by the pancreas and this also is in the duodenum. Right? And let's now go to that part. I think that the only part that is left is the function of the small intestine, which is absorption and assimilation. So Hamza, go through these two paragraphs and then I'll start with the discussion on villi. So just read these two paragraphs.
Right. Okay.
We're done with ingestion, we are done with digestion, and now we move on to
Sorry, miss?
absorption. I said we are done with ingestion.
Yeah.
And physical and chemical digestion. And now we move on to absorption. So absorption is done by this, some parts of the absorption are done in the small intestine and some in the large intestine, right? Now, let's first begin with the small intestine. That inside the small intestine, if we look at it through a light microscope or through an electron microscope, we will see that the small intestine is thrown into thousands and thousands of folds. And then again, those folds are thrown into further folds and further and further and further, right? It's like this. For example, I can draw a diagram to tell you. Let's say that this is your small intestine. It looks like this on the normal diagram, right? Now, if I enlarge it, it's folded. These are the obvious folds that you have. Now it has folds like these, finger-like projections of folds. And then within these folds also, let me draw even smaller folds. We have folds over here as well. Are you getting my point?
Yes.
So folds into folds into folds. And then, for example, if I pick up, what do? If I just pick up this, okay, and I zoom into this, which I'm doing it right now,
Yeah.
zooming into this. So I see that one fold looks like this under the microscope. But then surprisingly, if I also zoom into this, if I keep zooming in and zooming in and zooming in, so what do I see? I see that every single villus is again made up of what? Thousands of such cells, these cells. Basically, these cells are the epithelial cells of the villus. And if I just pick up any random cell, I'm just talking about this one little tiny cell, epithelial cell.
Yeah.
Even this tiny epithelial cell has folds. So just imagine.
So it's like a villi inside a villi in a way.
Exactly.
Right?
Yes. And you can also say that villi is villus is a very interesting structure which has folds which are macroscopic and which has folds which are microscopic, which means those folds which you can see with your eyes and those folds for which you need a microscope. In fact, these folds can only be seen with an electron microscope. Not even with the light microscope. With the light microscope, maybe you can see this fold only, right? So whenever you have such a structure inside your body, we use a term for this, and we say that in fact, now what I'm doing is that I'm making a table and I'm writing the adaptive features of a villus. What has the villus been supplied with so as to enable it to perform its best function, right? So I'm starting and you can take down notes. Hamza, are you there?
Yes, I'm miss.
Okay.
Can you hear my keyboard now? Or
Yes, I can, but it's okay.
all right. I'm just gonna quickly write down this and I'm gonna stop.
When you're done, let me know. second, and why are we calling it hepatic?
This meeting is being recorded.
Okay, so you can see, Hamza, in front of you that I have written the topic assimilation at the top, and I'm trying to explain you through this diagram of mine a stepwise manner of absorption of glucose and amino acids into the blood capillaries of the villi. Can you just first try to make sense of it on your own? And if you are stuck, and then I'll explain you.
This meeting is being recorded.
Oh God. Acha. Let's start with the fact, and the hard part is actually confusing you because you haven't done the structure of heart with me. So that's why I think I should not talk about the heart right now. If we completely ignore the fact or ignore the involvement of the heart over here, and we just focus on the point that, let's say you're done with absorption, you're done with everything, right? And now your body needs to make use of these substances by absorbing them into the capillaries of the villi. This is the villus, okay? This is a small chunk of the villus, which I have, just shown that glucose and amino acids are going to get absorbed into the villi, capillaries of the villi, and when they get absorbed, they will be carried by this vein, and the name of the vein is hepatic portal vein. Portal means a portal, right? It's a portal that is actually carrying amino acids and glucose to the liver. Because you see, the glucose and the amino acids are of no use to your body, even if they are absorbed by the ileum, unless and until the liver knows how to handle them. Okay? And this handling is actually assimilation. Even if you have digested, ingested, absorbed your food very, very perfectly, but if your body does not know how to handle it or assimilate it, it's useless. So glucose and amino acids will be carried by the hepatic portal vein to the liver. In the liver, all the excess glucose molecules will be converted to glycogen. All the excess amino acid molecules will be broken down. The amino group will be removed. They will be deaminated. Okay. And then whatever glucose and amino acids, they are needed by the body, needed. Okay. They will be carried by the hepatic artery. Let me just draw the hepatic artery. This is the hepatic artery. The hepatic artery, it will carry the absorbed glucose and amino acids to the rest of the body. Right? Now, is it understandable?
Yeah, it's much more.
Now you explain it to me.
Okay.
Step one.
So first of all, glucose and amino acids are absorbed by the capillaries. That's quite simple. Right? Then the capillaries make their way to the heart, right?
Okay.
Okay. Then that food or those nutrients, which are glucose and amino acids, they transfer into the hepatic vein. Right? And is the hepatic vein connected to the heart, miss?
You're talking about this vein?
Yeah, hepatic portal vein.
It's not connected to the heart. The hepatic portal vein is a connection between the gut and the liver.
Okay, so oh, is it the hepatic duct?
No, it's not the hepatic duct.
So
it's a vein.
It's a vein, right? Okay, so it's connected to the liver and the what, miss?
Hepatic portal vein is actually a connection. It's a bridge sort of between the gut. By gut, I mean ileum over here, right? Between the ileum and the liver. Now see, when the ileum has done its job, when the villi have absorbed glucose and amino acids, what will the villi do? They will send these absorbed glucose and amino acids by means of the hepatic portal vein to the liver.
Why will they send it to, then what will the liver do again?
The liver will decide like this. For example, the liver gets 1,000 molecules of glucose and 1,000 of amino acids.
Okay.
And liver knows what to do. Actually, for example, the liver thinks that only 10 amino acids are needed. Okay. So it will be sending.
And does it store the rest?
It actually, yes.
Right. So does it store them in the form of glycogen?
Yes. And it will never store amino acids. Liver will break down the amino acids by removing the amino group from them and causing deamination.
Okay, so it'll remove amino from them, which is through deamination or deamination, right?
Yeah,
So when the amino acids are deaminated by the liver, what's remaining?
urea.
Urea.
And that
Okay, and when
and it's that urea which will now be sent to your blood, and then that urea will be eliminated out of your body through the urine.
so means if we consume extra protein, right? So
Huh?
suppose we consume extra amino acids, or I've eaten protein today in lunch, and then I consume it in like during dinner again, and my body does not need protein. Suppose it
Huh?
doesn't.
Huh?
So then what will happen is amino acids. So digestion takes place. What do you call? Protein breaks into peptidoglycan. Peptidoglycan breaks into amino acids. The villi in the small intestine absorb the amino acids, send it to the capillary vein. The capillary vein obviously transports the things to the heart, right? And
Hmm.
midway through, it reaches the what do you call the hepatic portal vein, and
Hmm.
then the hepatic portal vein is basically the connection between the ileum and the liver. So it'll send the nutrients, which is the glucose, as well as like if I'm eating rice with protein, or if I'm eating suppose roti or bread, right, with what do you call protein, they'll send the glucose and amino acids all the way to the liver. What the liver will do is it'll store glycogen, glucose in the form of glycogen, right? So in the form of polysaccharides, and what, right? Is that true?
Correct, correct, 100%.
And then in from amino, well, and then with amino acids, it'll break them down by deamination, right?
Hmm. Hmm.
And and then it'll form the product urea, and urea will be transported to our what do you call
Urine,
large
kidney.
kidney, right? And then urea is formed when what do you call our kidney mixes it with water,
Exactly.
urine is, right? And then it excretes it through the anus. So what will happen to those amino acids which have been gone through deamination? Like, is there something remaining of the amino acids? Are they stored, or are they just completely all excreted out?
They are, it's like this. For example, if you have an amino acid looks like this. There's an R group, there's an H,
Yeah.
this is
There's
amino.
an
Hmm? Now, when you deaminate it, it means you remove this. Okay. And then you're left with this. So
Okay.
in this process, what happens? There are some other chemical changes which happen, and then eventually you end up with the formation of urea. So, so I would say that the end product of deamination of amino acids is urea only. And maybe, I think the carboxylic acid might be used for some reason. I'm not sure right now, but basically urea is formed. So this means that if a person has a high protein intake, it means his body will automatically produce a lot of urea. And if he is doing gym, or he is taking a lot of protein tablets or protein shakes, or high protein diet, then he should have a lot of water, because you need water to eliminate excess urea from your body. Otherwise, what will happen? The urea concentration in your blood will go on increasing, and that's not a good thing. Are you getting this?
Yes, I am. So
Yeah.
RCH and COOH. So carbon. What's that called? COOH.
Carboxylic acid.
Carboxylic acid, right? So when an amino acid goes through deamination, H,
Hmm.
so nitrogen and two atoms of hydrogen are removed from it,
Hmm.
leaving RCHCOH. Okay, and then those are stored in the liver.
No, they will be converted to urea. Since you are so much interested, so let me just show you the exact reaction. Just a minute. There we go. And there is the reaction. Hmm. Look, this is the reaction. In fact, this reaction is more clear over here. Look at this amino acid. The amino acid is
Yeah.
deaminated, and then you end up with forming a keto acid. And this keto acid, which this thing, we call this a keto acid, it is either respired, because it still has the carbon hydrogen. So it has some trapped energy inside it, so it can be respired, or it can be even converted to glucose or fat. And this ammonia thing, which has been produced because of removal of amino group, this ammonia will now combine with carbon dioxide, and then urea is produced. So this part
Okay, so
can be recycled.
you're saying that okay, I get it now. Right. Okay. So when the amino acids go to the liver, what the liver does is it removes the nitrogen and H2, right? And then
Hmm.
it turns it into ammonia. Ammonia is mixed with the carbon dioxide from the environment, and urea is formed, right?
Exactly,
So
exactly.
water, yeah, so urea is formed.
Hmm.
After that, what happens is that we're left with RC, oxygen, and then COO, right?
Huh.
COOH.
It's
And then this whole thing, which is a keto acid, can either be converted to glucose or fat, or it can be respired. So if respiration is needed, it can be respired. And respiration is not needed, then it can join the glucose to form glycogen. Is that it?
exactly. It can even be
Okay.
converted to fat. So you can see how, how perfectly your body tries to balance you. And even, it's just like this, that your body doesn't even waste a single molecule. We waste so many things in our life, big, big things, right? The body doesn't
Yeah.
even waste a molecule. This is what this, you can't even see it. A molecule is so tiny, so the body doesn't even waste this tiny molecule. It will either, if you need energy, it will be utilizing it for energy, and if you don't need energy, it will be converting all that excess of the keto acid into glucose or fat. So is that now understood?
Sorry, miss?
Is that understood?
Yes, it is understood. I get it now.
Okay, just a minute. Mm, yeah, here. So now this thing will be very, very easily understandable, right? Let's say, for example, if I read from over here, I'm reading the second paragraph, that most of these substances pass into the blood capillaries inside the villus. The blood capillaries from all the villi link up to join a vein known as the hepatic portal vein, which takes all the substances to the liver. The liver cells absorb and assimilate these substances. Glucose may be changed by the liver cells to glycogen for storage. Amino acids are used by the liver cells to make many different kinds of proteins, some of which are then returned to the blood to be transported to other parts of the body. And if there are more amino acids than the body requires, they may be broken down in the liver to form urea, which is excreted in the urine. Is that understood now?
Yes.
Let's just finish this chapter today. So that then in that case, we will be up to the mark that we decided. In fact, let's look at our timetable. This is
Miss, could you just go back to the previous page and give me like 5 minutes around to jot stuff down quickly?
this one.
Yes, please.
Acha, okay. I'm waiting. That
This meeting is being recorded.
what's the function? What's the function of the villi? So the villi hugely increase the surface area. This is how you will write your answer. The villi hugely increases the surface area of the lining of the small intestine. Why? Because every villus is again covered with thousands of microvilli. So the total surface area is even increased more. What connection does it have to the function of intestine? The more the surface area, the faster the rate of absorption. Okay, so you will write this down. The more the surface area, the faster the rate of absorption.
Okay, miss.
Let's see how much is left. Okay, so we are done with this chapter. Let's do some questions quickly. MCQ number 1. What's the answer?
What color is going to be lack of vitamin? Lack of vitamin C in the diet.
Correct. Number 2.
When the digestive system or protein enzymes secreted, stomach and pancreas and liver and mouth, or protease enzymes. So I believe they're in the stomach and pancreas, miss.
Correct. Number 3.
Most water absorbed in the blood. Most of the water is absorbed in the small intestine.
Let me see. In some books, it's written small intestine, and in some large intestine. I think so. Large. Did we read about large intestine in your chapter? Just a minute.
Miss, I read it by myself, right? And in the notes I did,
Mhm.
it said that 80% of the water is absorbed in the small intestine, along with the nutrients.
Really?
Yeah, I don't know though, because like you said right now, some books say large intestine, some say small.
Okay.
So I don't really.
Okay. I think your book is saying small because it's written a
Yeah.
smaller quantity of water is also absorbed in the colon. So colon is a large intestine, right? Okay. So then this means that the answer is small intestine. Okay. What is assimilation? Do you think assimilation, you just read these two options and then I'll move on to the next page. Do you think assimilation is A or B?
The movement of small molecules or the transfer digested small molecules into the blood. So it's B.
Let's see C and D. The movement of small molecules into cells where they are used.
Yes, this is used. Yeah, that's assimilation. Yes.
C. What is the function of bile?
Emulsifying fat. So B, miss.
That's emulsifying proteins.
Oh, sorry. Chemically digesting fats. No, that's not that. Oh, increasing pH.
Increasing pH. So this question is a little bit confusing. Now the diagram shows the digestive system and quickly tell me the names of these parts. Okay, so what's A?
A is the salivary gland, miss. I think.
Correct. What's B?
The esophagus.
Okay, what's C?
The stomach.
J.
The liver.
D.
Pancreas.
E.
E is the duodenum.
F.
Small intestine.
G.
Large intestine.
H. Colon.
The rectum.
Rectum, sorry, not colon, rectum. And then I is the anus. Okay. Let's do this. The diagram show the teeth in the upper jaw of a human. Name the types of tooth labeled A, B, and C. What's A?
The canines.
A are just lying down in the front, so they have to be incisors, the cutting ones.
Oh, no, not canines. Sorry, incisors.
And what's
I
B?
messed up. B are canines.
Canine. And then what's C?
They can either be molars or premolars,
So,
but they look like molars to me, not premolars. They don't look like premolars.
molars. Okay. Let's just skip this and in fact, I'll go to this book. I still remember I had done some questions. Yes, there are lots of questions over here. Okay, let's do this one. What is K?
Wait, what's this? Oh, this is the teeth. Tooth. Okay. K is the dentine. No. Enamel. Sorry, enamel.
Achha, what is L?
I believe that's the dentine, miss.
Okay, and what's N?
They're the blood capillaries.
Basically, the pulp
The
cavity.
pulp.
Huh.
Sorry,
And what's
the
M?
pulp. M is, I'm not sure what this part was. Maybe the gum? Or no, this is the bottom part.
The root.
The root. All right.
Right, the root.
Miss, the root wasn't mentioned.
Yes?
The root wasn't mentioned, was it?
I also think it wasn't, but okay, fine. Basically, see, every teeth has a part that is embedded and the one which is visible, right? So the part which is somewhere, which is lying below this point is not visible. So this entire part is the root, actually. Okay, never mind. Maybe this is a question from some old past paper. Describe how dental decay is caused. So we've done this. I'm skipping this question. And okay, so what can be the answer to this question? B, part 1. State a possible effect on the fetus of a shortage of calcium in the mother's diet. What will the fetus have? The problem that it will have?
Fetus will have weak bones.
Rickets?
Yeah, no, yeah, I believe it will be rickets.
Rickets, right? Okay. And there is...
So
yes, you want to say You want to say
Yes, I
something?
was saying rickets is formed when there's a deficiency of vitamin D, right?
And
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 vitamin
and calcium.
and yeah, so vitamin D is simply like the loss or when sorry, when calcium isn't absorbed in the blood
Yes. Yes.
or sorry, the bones.
Figure 8.1 shows the structure found in the wall of the small intestine. So name this structure.
This is a villi or
State one.
villa, sorry.
Same thing. State one function of this structure.
Absorption.
If I was an O-level student, I would never simply write the word absorption. It's better to write
Yeah, the function of this is to absorb the nutrients.
nutrients, and it's better to write down glucose and amino acids, right? Acha, okay. This table contains information about the digestion of three main types of food. So fill in the spaces. Yes, what will you write?
Food type, starch, enzyme. So starch is what do you call amylase? Fat is lipase, right? And the products of digestion will be glycerol and fatty acids.
Correct. What
And
about
for
the last one?
and last one is it's going to be amino acids.
Okay. Now this is your favorite question. This should be your favorite because we had a discussion on this so much. So what will you write as far as the answer to this question is concerned?
The two ways in which the liver may deal with these products of protein digestion. The liver may deal with these. Okay, so what'll happen is two ways it'll deal with it. Okay. So the first way is that it'll remove the NH2 from the amino acids, right? Which will form and mix it with what do you call carbon dioxide to form urea and
Correct.
send it to the kidney. The second way is that it'll the second way is that it'll simply respire or store it, right?
No, wait. Products of protein digestion, two ways in which the liver may deal with these products of protein digestion. So simply say one way is that it will convert it into urea, and the second way is that it will use those amino acids, send those amino acids to the body if they're needed to make proteins.
Yeah, right.
Right? Acha. November 15. Did we do peristalsis in this chapter? I don't think so. Was there any mention of peristalsis in this chapter? I think it has been. I think so. It has.
Like the wave-like contraction in the esophagus, right, miss?
It was not there in your textbook because if I go back and look at your syllabus, let's just check. Okay, since we have finished the chapter, so let's just have a check on whether we've missed out on anything. So wait.
I'm not sure.
Wait a minute.
Aren't peristalsis simply like wave-like contractions that force the stomach? That forces the bolus to travel down.
They are, but somehow it seems to me that they have removed this learning outcome. See, we just give it a check. Have you done all this, Hamza? Give it a check. Let's go through this from 1 till all parts 1 and all parts 2.
Identify diagrams, images, the main organs of digestive, to elementary colon, mouth, esophagus, intestine, duodenum, ileum, larynx, colon, rectum, and anus. I've done that. Associated organs, salivary gland, pancreas, liver, gallbladder. I've done that. Describe the functions of the organs in the digestive system. Ingestion, digestion, absorption, assimilation. We've done that, right, miss?
Yeah.
Miss, it says ingestion, digestion, absorption. Okay, never mind. Yeah. Okay. So I was just thinking of chemical digestion and physical or mechanical digestion, which is simply the two forms of digestion. Right, we've done this.
Achha, let's move on to the next page. Have we done this? Go through this as well.
Physical digestion is the breakdown of you. We've done that. State the physical digestion increases. We've done that. Identifying diagrams and images, the types of human teeth, incisors, canines, premolars, and molars. Yeah, we've done that as well. Describe the structure of human teeth related to an animal. Describe the structure. We've done the structure, but we haven't done the functions of them, right? I don't think that's my level yet.
Outline the role of emulsifying fats and all. And then let's move to this also. Let's keep a, make a checklist whether we've covered this or not. So go through these also one by one and let me know.
Describe chemical digestion. We've done that. State the role of chemical digestion in preceding. We've done that as well. Describe the functions of enzymes as follows. Amylase breaks down starch, proteins, and lipids. We've done that. State where in digestive system amylase, proteins, lipids are secreted and where they're active. We've done that. Describe the function of hydrochloric acid. Hydrochloric acid in a gastric juice limited to a harmful organisms and food providing acid appeared for the enzyme activity. We've done that. Describe the digestion of starch in the digestive system. Amylase breaks down starch into maltose. Maltase breaks down starch glucose in the membranes of the lining. So we've done that. Describe the digestive proteins. Trypsin breaks down protein in acidic condition. Trypsin breaks down protein in alkaline condition of the small intestine. So Miss trypsin is what do you call found in the duodenum, right?
Hmm.
Okay, and it is simply secreted by the it is simply secreted by the pancreas, the enzyme itself.
Yes,
Trypsin.
yes. And actually, you
That's
know
not
what? If you remember, today only I told you this, that although the action of both these enzymes is the same, right? But the environment is different. The optimum pH for pepsin is acidic, and the optimum pH for trypsin is alkaline. Pepsin
okay.
will do incomplete digestion of proteins, meaning it will convert proteins to polypeptides, whereas trypsin will further break down polypeptides to proteins. So this you have to remember because this is one of your learning outcomes.
Okay, and Miss, can what do you call? Can trypsin break down
Proteins?
break down proteins into peptidoglycans?
Why did we use the word peptidoglycan? Peptidoglycan is actually a combination of proteins and polysaccharide. Peptidoglycan is actually the composition of cell membrane of the bacteria.
This is a peptidoglycan?
It's peptidoglycan.
What about the word right now? So proteins breaks down.
Polypeptide. Polypeptide.
Oh, polypeptides, right? I keep mixing it up. Okay.
Never say peptidoglycan. It's a completely different thing.
Okay, Miss. Right.
Right?
So polypeptides.
It's like this.
Okay.
For example, you know, if at a very simple level, if you want to know now, let's say that these four shapes are different amino acids, and I have joined them together to make a polypeptide. Why do I call it polypeptide? Because it has these peptide bonds, 3 peptide bonds. Okay, polypeptide. Achha, many polypeptide chains, if I have another polypeptide chain, they will become what we call a protein. So now in the stomach, what happens is that in the stomach, pepsin is going to break down protein into polypeptides, meaning stomach will only do this digestion. Let me remove this, okay, like this. Are you getting it? And then this job will be done by pepsin. Now, what is the job of trypsin then? Trypsin will convert polypeptides to amino acids. What do you mean by that? Trypsin is going to break these bonds. Now, are you getting it?
Yeah, Miss, I do get it. But my question is, can trypsin break down the proteins?
Yeah.
Yeah.
It can.
Okay. So trypsin can break down proteins to polypeptides, and it can break down polypeptides into amino acids, right?
Exactly.
However, pepsin can only break down proteins into polypeptides.
And pepsin, you will use the word pepsin only for the enzymes in the stomach.
In the stomach, right?
You don't find pepsin anywhere else besides the stomach.
And you find what do you call
Trypsin.
Trypsin. Do you find trypsin anywhere else? So in the stomach?
No, you find it in the intestine. You find it in the duodenum. You might find it in the ileum also.
Okay. So the ileum and
duodenum.
Duodenum, right? Yes. And in ileum, they are secreted through the pancreas.
Exactly. And always remember that pancreas is a leaf-shaped organ which is surrounded by the C-shaped curve of duodenum. Okay. And the pancreas is producing lots of enzymes. And right now in the middle, which you can see, which I'm actually highlighting, is the pancreatic duct. Now, once the pancreas produces this enzyme, it's going to pour these enzymes through the pancreatic duct into the duodenum. So this means into the duodenum, the pancreas is going to pour what? It will pour pancreatic amylase, it will pour pancreatic lipase, and it will pour pancreatic trypsin. All 3 enzymes. And it is these 3 enzymes that will sort of, you can say, they will complete the chemical digestion. They will complete it.
Okay, so just let me get this straight once more. The
Yes.
only enzymes that are produced in the membranes of the epithelial cells of the small intestine are maltase, right? Those are the only type of enzymes produced. All the rest of the enzymes either are secreted through the pancreas, are only secreted through the pancreas, and duodenum itself makes no enzymes, right?
Duodenum can make its own secretion. I'm not 100% sure about the duodenal enzymes, but basically the enzymes, they come from pancreas. Even if some of the enzymes are made in the duodenum, but that's none of your concern right now because we don't teach digestion in so much detail. Digestion is itself a very, very detailed topic, right? So we can't, go beyond the scope of your syllabus. It will be too much for you.
Okay,
Hmm.
I get it. So duodenum is none of my concern.
Yes. And
So all that,
if we go and again we check, we also covered this learning outcome that bile is an alkaline mixture whose job is dual. It has dual job. The first job is that it is going to neutralize the acidic mixture of food and gastric juices entering the duodenum from the stomach so that it provides a suitable alkaline pH. And the second function is emulsification. Because you see, many students think that the only job of bile is to emulsify fats, but there is another job also, which is to neutralize the acidic mixture. And that was the question that was given in the MCQs. Remember, which we
yes,
just did right now, right?
yeah. So, so what do you call it? The pH level of bile is higher than 8, right? Because the pH level of the stomach is 2, if I'm right, and the pH level of the small intestine or duodenum is around 8, right? So the
Correct.
pH level of, just for extra information, the pH level of bile must be 10,
Exactly.
right?
Exactly.
So it must switch from 2 to 8. So there must be a difference of about 6 or something.
Of course, of course.
Okay, that makes sense.
Okay, so I think we're done for today. And if we go
Yeah.
and check this now, Hamza, just a minute. Let's try to reward ourselves basically that we've accomplished something. So this is the textbook and this was our plan. So let's go to the first page. Just a minute.